Okay, so now we proceed to production of insulin using cDNA. Okay, so firstly, mRNA that codes for insulin is extracted from beta cell of pancreas. Okay, extracted here, dia membawa maksud yang sama dengan isolated. So, mana kita nak ambil mRNA that codes for insulin? Actually, it is extract or isolate from the beta cell of pancreas. Kenapa beta cell of pancreas? Sebab secara normal, beta cell of pancreas will secrete insulin. So, that's why mesti kita perlukan mRNA that codes for insulin from the beta cell of pancreas. Okay, and then after that, mRNA will undergo RNA processing or RNA splicing to remove intron, producing a functional mRNA. Okay, so RNA processing or RNA splicing is important because of what? The mRNA strand, kalau kita tengok based on this diagram, okay, so this is the strand of mRNA. So actually before mRNA is undergo RNA splicing or RNA processing, we call that as pre-mRNA. Okay, so kalau awak tengok dekat sini, dia ada exon and also intron. So, yang kita perlukan hanya exon. Intron tak perlu. So, that's why bila berlaku RNA splicing, so it will remove intron. So, that's why once RNA splicing is already occur, so it will produce functional mRNA that consists only exon. Okay, so next, mRNA is used as a template to synthesize first strand of cDNA. Okay, so let's say here, kalau kita tengok dekat sini, so this is the mRNA that contain only exon. Okay, so this mRNA will use as a template to synthesize cDNA. So how the first strand of cDNA is produced or is synthesized? By using enzyme reverse transcriptase. Okay, and then once first strand of cDNA is already synthesized, so what happened to the mRNA? mRNA is then degraded by using mRNA degrading enzyme. Okay, and then next, DNA polymerase is added to synthesize second strand of cDNA complementary to the first strand. Okay, so kalau kita tengok based on the diagram, okay, kalau tengok di sini, okay, so this is the first strand of the cDNA. Okay, so once first strand of cDNA is already synthesized, so what happened to the mRNA? mRNA will be degraded by using mRNA degrading enzyme. So, daripada first strand of cDNA, how it will synthesize the second strand of cDNA? Actually, it is by enzyme DNA polymerase. Okay. So, bila ada DNA polymerase, so that's why if you look at this sentence, DNA polymerase is added to synthesize second strand of cDNA complementary to the first strand. So, once the second strand of cDNA is already produced, so it will produce double-stranded DNA. Okay. And then, what happened after that? So, kalau awak tengok dekat sini, starting from this point until the last point, actually, this is the steps in gene cloning. Maksudnya, yang macam saya explain awal-awal tadi, yang tu lebih kepada how mRNA is isolate, okay, and then how cDNA will be produced. So, bila dah terbentuk cDNA, so cDNA and plasmid are cut by using same restriction enzyme. So, actually cut here, it is the second step in gene cloning. Okay, so once cDNA and plasmid are cut by using same restriction enzyme, okay, so it will produce complementary sticky end. So remember, okay, yang akan hasilkan sticky end adalah restriction enzyme that we call as EcoR1. And then the cDNA is inserted into the plasmid and joined by DNA ligase, forming recombinant DNA. Okay, then recombinant DNA is transferred into bacterial host cell by transformation. Okay, so once recombinant DNA is already transferred into the host cell, so amplification process occur. So if you look at here, the last step in gene cloning, so blue-white screening occur to detect host cell containing recombinant DNA. So apa yang awak perlu take note untuk production of insulin using cDNA, maksudnya awak mesti kena tahu macam mana daripada mRNA, will produce cDNA, and then after that, actually cDNA tu sebenarnya adalah kita punya target DNA ataupun gene of interest. So once kita dah ada kita punya target DNA, and then kita ada plasmid. Plasmid ni adalah adalah DNA cloning vector. So both cDNA and plasmid are cut by using the same restriction enzyme. So maksudnya step yang seterusnya sampailah habis. So actually this is same as the steps in gene cloning.